I'm very pleased now to introduce you to Richard Hullihen, who is the Chief Executive of an investment that we have in the portfolio called Polarean Imaging. And this is a company we've been invested in for a couple of years now. It's a very promising and important position in the VCT, so I'm really thrilled to have Richard with us to try and explain it to you in a little bit more detail. Now, unlike quite a number of the other interviews we've been doing, Polarean is it's selling a medical technology. These always have their complexities, so we're going to do our best to really try and put over to you something of how this works and what it does and why it's important. So that in essence, what the kind of images we're going to be talking about are all lung images. And the reason we invested in Polarean was because we think they've made their imaging represents a significant step forwards in lung imaging and the ability for physicians to diagnose lung diseases through having available much more accurate, much more detailed, more powerful imaging capability. And I understand from the company and from, as a non-scientist myself, but what I picked up is that some of the technologies that are still used in lung imaging are very old, and there haven't really been many advances in this area for a long time. So what Polarean has is particularly exciting because it's the first real innovation in this area in quite a long time. And we're going to talk more about the actual equipment and its history later on. But first of all, I think it would be good to introduce you to Richard and jump right in with some discussion of the actual images. So Richard, it'd be great if you could maybe talk us through a few of the images we're going to have a look at and give us a flavor of the kind of output that the Polarean's technology gives.
I would be happy to do that, Paul. Thanks very much for the opportunity, and pleased to meet everyone today. Thank you. So I think if you look at the image which Paul is displaying, which says from qualitative to quantitative xenon ventilation imaging. So the history of medical imaging, as you probably would well know from having been a patient in a hospital at some point, is the use of X-rays to do things like chest films, which were the most ubiquitous medical imaging procedures on the planet circa 1970. Those images and CT after them were all structural or morphological images. So X-ray was simply a two-dimensional presentation overlaying all of the soft and skeletal structures in the body into a single two-dimensional image, which was kind of the origin of radiology. That was interpreting two-dimensional images representing three-dimensional structures. CT, to a great degree, was invented to unravel that problem for medicine. And so in the case of both the head and the thorax of the body specifically, those technologies finally presented them in high-resolution 3D representation, simplifying the diagnosis of many different kinds of problems. At the end of the day, however, those things were about structure, and much of what plagues medicine today is about function. So it's not where is something in your body, it's about what is it doing and how is it working. And so our technology fundamentally is to explain and quantify and display pulmonary function. And we do that by using an inert noble gas as a contrast agent, Xenon 129, which is inhaled in a simple 10-second breath hold exam, which requires no effort on the part of the patient. You simply breathe it in. We take the image very quickly in the bore of an MR machine. It is literally the fastest exam in an MR machine. It's only a few seconds. It takes longer to get the patient in and out of the machine than to collect the data. And from the data that we collect in that scan, we can now assess not only ventilation, which is simply how your lungs inflate, remembering that your lungs essentially look like an upside down tree with 23 branches of airways going down to a microscopic level. And then having illuminated ventilation, we can answer the most important questions in pulmonary medicine, which are how are your lungs transiting oxygen into your arterial blood flow? So in order to do that, if you remember your high school biology class, you have an alveolar structure, tiny sacs, which have very thin membranes where this magic part of being a mammal happens. Oxygen goes into arterial blood, CO2 comes out, and the oxygen is then carried away by red blood cells to do its duty in fueling the body. So we can quantitatively assess gas exchange at every state in the pulmonary process and provide that information simply in color-encoded images to the pulmonologist and the radiologist, and even to the patient to show quantitatively how that process is working.
So thank you. And I'm just going to maybe dumb this down terribly just by pointing out that pulmonology is the science of the lung, right?
Exactly. Yes. Sorry. We live there. We use, I'm hopefully don't let me use any acronyms, but yes, pulmonary medicine is the study of the function of lungs. Correct.
And so I was very struck when I first saw these, that picture right at the bottom right there, this colorful three-dimensional, I guess, image showing through the lung contrasts pretty fantastically really with what you would have otherwise seen, a black and white image, not showing you about anything about lung function, but just showing you maybe which areas of the lung in general weren't working.
Yes, no, the lay explanation is it lights your lungs up like a Christmas tree. When we hyperpolarize xenon in our equipment, it creates a 100,000-fold increase in its signal level in the lungs. It's astounding. There is no analog for this in medicine. Medicine in many cases is about the detection of contrast differences in images. And this dramatically simplifies the detection of defects in the lung. It's just a wonderful thing.
Yeah. Now, I understand that when you were discussing with the FDA, the regulatory authority in the States, which we're hoping will grant approval to this technology shortly, how to prove the worth of this, what they suggested was that you did an equivalent study or a non-inferiority study with what they regarded as the previous standard, which was radioactive scintigraphy, which also uses xenon. Do you just want to say a little bit about the difference between radioactive scintigraphy and this
Yes.
type of imaging?
Sure, I would be happy to. So it turns out that xenon is the element in the periodic table with the highest number of stable isotopes, meaning the number of variations in its elemental constituents, all of which are xenon. It turns out that one of the neighbors in the periodic table is xenon 133. We are xenon 129. It just so happens that xenon 133 is radioactive and has been used as a contrast agent for about 40 years now in a very limited way in nuclear medicine. Of course, nuclear medicine is the part of medical imaging which requires that the patient either inhale or be injected with a radioactive contrast agent. But it has some limitations. It's radioactive, so if you're going to be doing imaging recurrently to assess people, not a good tool in that regard. Secondly, it's two-dimensional, not three-dimensional, so you get a slice of the lungs as opposed to our natively three-dimensional acquisition. But nonetheless, it is approved. And in the way that regulators work, if you can prove your equivalence to something else that is approved, then you are then, by the associative properties of regulations, approved. So that was our assignment for our clinical trial, actually.
And you had the data readout, and it was a pretty simple yes or no kind of readout, I guess.
Yeah, that's the beauty of a head-to-head trial. It is quantitative arithmetic. You either won or you did not. And so we met our burden, the top line readout. And if you remember, the structure of our trial or the requirements of regulators is you just to make sure, and this is just kind of prudence at work, you have to do it at two different sites. You can't be vulnerable to a single site having a bias. And you have to use two indications, two different clinical questions that have to have an answer that change the course of care for a patient. So our trials were done in the case of patients destined for lung transplant or patients destined for lobe resection surgery as part of a cancer therapy. And so we did that. We had sets of patients at two different sites. One of our largest sites, the University of Virginia and Duke University, which is our home base. The trials went uneventfully. We had no significant adverse events attributed to our drug. We completed it, and the top line readout was as we expected. So that was great. It was just the best possible outcome.
Fantastic. So if we just have a now look at the next image that you've given us to discuss, this I understand shows the image looking at three different aspects of lung function.
Yes. So ventilation is one part of pulmonary function, and you would most commonly know it. Virtually anybody, I know I do in our extended family, as somebody walking around with an inhaler in their pocket. And so in the ventilation case, if you can't breathe, you use an inhaler, your airways are dilated, and your ventilation problem might be solved. But the other cases of pulmonary function are more insidious and have to do with the function of your alveolar tissue in transiting oxygen into arterial blood flow. And so that's barrier tissue or alveolar tissue. And then finally, RBC or red blood cell transport, which is taking your oxygen away through the cardiac cycle to the rest of the human body. We happily, because of the MR physics of this, are able to quantify our drug agent, our contrast agent, at each of the steps in that process. So out of the single scan, we get three different pieces of information. One is ventilation, one is barrier transport, and the final is red blood cell affinity. So we can answer for the pulmonary physician any question they might have about what's going on here. And the interesting thing that's being discovered is that each of the various forms of pulmonary disability that exist wind up having characteristic sets of images explaining their behavior, which is very exciting going forward.
And has it been possible with former imaging technology to know anything about what's happening in the barrier or the red blood cells?
Yeah, it simply isn't. It's not able to be assessed. Essentially what happens is people empirically evaluate drugs using endpoints like 6-minute walks to answer questions, which explains why drug development is so expensive, why clinical trials are so huge, because the statistics of these kinds of endpoints in trials are so noisy that you have to have impossibly large bodies of people to understand whether you're having an effect on something or not. We're the only way to non-invasively assess this quantitatively, which has a lot of people very excited because it could make clinical trials, even phase 2 trials, let alone phase 3 trials, much smaller. Right?
And can we just talk a little bit now about the actual equipment that's involved in this?
Yes. So
there's a number of different parts, and actually these have a very long history, some of these, which I always find interesting.
Yes.
In development for something like 20 years.
Yes, they do. It's quite a story. It could be a novel someday. So this technology began with the investigation of a different noble gas, helium-3, which it turns out was only useful for ventilation imaging because it's not soluble in blood. It was then shifted to Xenon 129, which is soluble in blood and has all of the benefits that we just described in the downstream indication. But at the time that the original development program happened, a couple of things were true. One was, as everyone probably knows or would recall, the scan times for MR systems were 20 or 30 minutes long, which of course makes it impossible for assessing pulmonary function. No one can hold their breath that long. The second thing that was true was just the situation at most of the regulators who had characterized their approval process as either being one for a drug or one for a medical device, and didn't conceptualize the existence of a technology set like ours, which bridges between the two. We are what the regulators call a drug-device combination. And so in the case of a pure drug trial for clinical trials, this technology, if it had been commercialized initially, would have faced, hundreds of millions of dollars in trying to get approved because of the former conversation on the endpoints for trials. So it was shelved by its original developers, which happened to be GE and Amersham. So along came breakthroughs in MR technology and breakthroughs in regulatory structure and approval which then rendered this possible. And so our CTO and founder went back to GE and said, look, the R&D team is disbanded. Nobody else would be able to do this. Why don't you give it back to us and we'll commercialize it? And they actually, to their great credit, said, yep, good, go ahead. And so we did. So we have a license agreement with them, which post-commercial launch, we will be executing, but it's a classic kind of academic level license. We're very happy with that. So that took us forward into peeling the onion now, most of which has happened because we've driven levels of polarization higher and higher. When this began, people were very excited to be able to polarize something at the level of 10%. We're now pushing 50%, which means we can do these exams with 250 milliliters of xenon gas, which is very easy for patients to tolerate, and yet brings all of this incredible information in a single scan.
So somebody wanting to use this equipment, obviously they need to have an existing MRI scanner, so they're not buying that from you, but they're buying a number of little bits of kit separately. So what you were saying about the hybrid model is very important for the business model here, where we have a consumable, which is the Xenon 129, which is going to be the drug in this case, but we also have a number of bits of hardware that you need. Yeah. Just talk us through.
Yeah.
It's important.
So polarization is achieved by essentially pumping the xenon gas in the presence of some elements to excite it. And so that's done with, as you might imagine, with a big laser in our polarizer. So the polarizer is a bit of kit that we provide. Then the FDA, and we think most regulators will view that we're manufacturing a drug at the point of use. So there's also a small QA station which simply certifies that the drug has been polarized and gives us a place to park it, a small environment to park it, which is beneficial in the clinical setting because that lets us segregate the workflow of producing the drug, which takes about 15 minutes, from the activities associated with getting the patient entered into the MRI machine's computer, getting them into the machine, getting them positioned, and all the rest of those kinds of things. Wouldn't want that to be an orchestrated act that had to be synchronized or anything. So lets us decouple those two workflows. And then once they're in the bore of the machine, we administer the drug. 10 seconds later, we get these images. So we get to sell our drug, which is a consumable on a per patient basis, the drug the drug dispensing device, which is an impermeable gas bag with a mouthpiece used on every patient, then the QA station downstream. And there is also, because MR is a radio frequency based technology, there's no x-rays in an MR machine, but we do obtain the signal by listening with a coil to the molecules in the human body resonate. And so there is a coil tuned to the xenon frequency that is also part of our sales kit. So polarizer, QA station, coil, consumable dispensing, and the drug. Right?
Yeah. And so just to give a sort of an idea to our investors about how the commercial development of the company works, so far the company has already made sales of the equipment for research use, in which case you're selling all those bits of hardware, but you're not able to sell the drug at a profit because it's not approved yet. But you have actually made a significant number of sales over the past few years of the equipment itself.
Yes, we have. We've now reached a scale. We have 25 systems out there. We've now reached a scale which is happily one large enough that allows us to engage having some strategic discussions with some pharma guys, because we're big enough now that we could answer small structured questions for them at handfuls of sites. So very excited about that, actually. The way that this works is academic institutions are able to acquire the technology. It's used for research, which means they can't bill anyone for it, insurance companies or the government. But at the end of the day, they're able to do research. And so that's turned out to be wonderful. We have built and shipped between 3 and 5 machines for the last several years, actually, and that's holding up quite well.
And these are not small purchases, are they? What's the price of the?
Yeah, no. So all in for the kit, it pushes $500,000 in the U.S. for these places. Most of them are obtained in grants. So there are S10 grants with the NIH for exploring applications, for example, in pulmonary medicine. And so that's where most of the folks get the money to do this. There are a few places that just spontaneously acquire the equipment because they see the benefit of research. And so they're actually launching programs to investigate the application of this with or without the grant. So we get those both ways. We have equipment in the US, Canada, Germany, and the UK.
And do you get the impression that this quite extensive research use is creating some pent-up demand for clinical use?
Yes, I think a lot of, as you would know, on the front end of most inventions is a lot of scientific noise, right? Where just because you can do something doesn't mean that you should. But we're now answering some very important clinical questions. So, for example, we did have a $3-year, $3 million grant with Cincinnati Children's Hospital on the application of this technology to cystic fibrosis. And the development of its use as a biomarker for CF. And that grant is just now coming to a close. A lot of people very excited about that. And we have, I think, the last count, over 1,000 peer-reviewed journal articles, which makes this all very real. It's kind of moving from science project into clinical medicine now.
Yeah. And just rewinding to what you were saying about the pharma companies who are getting interested in using this for assessing their own drugs. I think you have another image here, which maybe gives a flavor of that.
We do. There is specifically, I think, slide 6, which is the consequence of one of the manufacturers of a drug for treating idiopathic pulmonary fibrosis. So IPF is a subset of the pulmonary disease state known as interstitial lung disease. And there are a set of these diseases. IPF is but one. And the key question here is, drugs are being developed, but they have the attribute that many pulmonary drugs, in fact, many drugs in general have, which is they work really well, but not for everyone. And so the question is, how do you select patients to be put on these therapies? And then how do you prove that a patient is actually benefiting? So, much of what goes on in pulmonary medicine is literally handing patients plastic cards with 7 degrees of smiling faces on them, right? Do you feel good or bad, better than yesterday or not, better on this medicine or not, and those kinds of things. It's just staggering, the empiricism of how this all unfolds. Here we have a case of a patient that was first chosen because they were a responder, and then put on a 3-year surveillance regime trying to show the benefits of this drug. And it was definitive, right? We found the patient was a responder and then proved that over the 3 years, their barrier tissue performance is nearly normal, right? Well, this is hugely important because the only other therapy for this patient is a lung transplant. There is no cure for IPF. So you can also see that the pharma company then took that and drilled it right down to the lab pathology level of looking at the tissue as well. So this is an end-to-end demonstration of our ability to assess function in the presence of a drug therapy. And this is really important because there are several of these kinds of drug therapies coming to market across pulmonary medicine. And the question of how to find the right patient and prove that they're benefiting is otherwise an almost insurmountable problem for these guys.
Yeah. And it's clear from what we've been saying about the business model here that the approval of the product is absolutely crucial, because once we achieve approval, once we see the approval come through, then you can start selling this as a commercial proposition and charging for the drug, and the clinicians themselves can charge for the service.
Yes.
So at what point are we expecting that? And do you think will there be any delays in the FDA because of COVID-19? Or
I think to the best of our knowledge at the moment, not. So the FDA has almost seamlessly transitioned to this kind of remote environment. So we're having our calls with them. Our pre-meeting was a virtual meeting. Normally, these are face-to-face meetings. Historically and conventionally, you go sit in a room with these guys. which is beneficial for us because you get to read people, and it's much harder in the virtual setting. And they're not presently allowed to participate in Zoom meetings with the outside world. So their side is collaborating on a Zoom call internally, but we only have audio access to them.
Oh, really?
That's on, I guess that's on purpose at the moment. Maybe it will change over time. But at the end of the day, we had our call. We sort, as you know, with regulators, those things are all highly scripted. They get to ask questions prior to the meeting. So did we. The meeting itself is taken up then with the answers to those questions going back and forth and any dialogue associated with them. But we heard nothing in the meeting that has deterred us from our existing timeline. So we are now proceeding with the building of our submission according to the outcome of that meeting, and are, as far as we know, happily on track for that third quarter this year.
So you'll submit in the third quarter, and then how long do you think it will take to hear back?
Yeah, so historically, the specified time periods for this are 10 months for the drug approval, and an additional 2 months, because if you remember from the pre-story here in our roadshows, we're a drug, and so we're entitled to petition for Hatch-Waxman protection for the drug, which is a grant of 5 years of exclusivity in market. And so we are submitting for Hatch-Waxman, which gives them the additional 60 days. So in theory, 12 months from submission. Now we also, although we're technically, from a regulatory point of view, not entitled based on the structure of our clinical trial, we are nonetheless going to suggest, is probably the best word, that they consider us for fast track review because of our potential use downstream in studying the patients coming out of this. Whether they respond to that or not is not knowable, but it doesn't cost us anything to ask.
Yeah. And so broadly, if we just sort of raise the issue of COVID-19, how has that been impacting the business?
For us, kind of somewhat surprisingly, not very much. So on the one hand, we are working remotely. We're working at home as an enterprise. But at the end of the day, not too many people were going to work to put their hands on things. Some of the R&D guys do, but we actually had outsourced manufacturing, so we're not building machines in our business anymore. Happily, our suppliers are essential businesses or located in places that weren't locked down. So we've been able to continue building machines unrestrained. We're able to do R&D. We're doing all the things that we said we would in our presentations to investors. So we've begun software development as we said we would. We've begun doing extended developments on the polarizer itself, as we said we would, all without any tremendous effect. Maybe a little bit from an efficiency point of view, but we're still making our milestones and deadlines, so very happy about that. We also, I think on the commercialization side of things, that's actually almost a Zoom business by definition anyway. So no effect there. We have initiated several of the activities that we said we would. You know, the commercialization side of this is about turning perception into fact. So this is actually hiring specialists in fields to go answer questions that we had derived answers to kind of empirically. That work is underway, so very happy about all of that. So unchanged with respect to effects on our commitments and milestones in presentations to investors.
And will the likely cancellation of medical conferences limit your ability to kind of get the news out, the message out there to the...
Yeah. So happily, a bunch of that, it'll be interesting to see. So we will do the things we usually do. So our research guys will be publishing the same posters, presenting the same papers that they would have, just virtually instead. We'll be a little bit sad that we won't have a booth at the shows to actually meet people and talk about things. But in the meantime, we're meeting people and talking to people all the time anyway. So we'll see how the conference thing plays out. I think it remains to be seen what effects that has. It could be that they're perfectly effective vehicles for doing these kinds of things. We'll see.
Yeah, I guess there might be online conferences, or some people have suggested that in some doctors may be more amenable to more sort of direct communications because of the lack of conferences.
Yeah, it could be. I think a couple of them will have, the MR one is coming up here shortly. It was actually delayed once and then converted to an electronic one. It'll be our first kind of experience with that. We'll see how it goes. It's fairly well known. We know all the players. I think it's kind of our home base compared to the newer ones, the clinical medical conferences we've been going to. We're still kind of the new kids for those things, but so far so good. I think it will be interesting. It'll be different, but it looks like it should work. So we'll see how it goes.
Well, I guess some of the exciting research that some of the institutions who bought the equipment were doing is going to be put on hold for a while.
Yeah, right now, I think until the institutions themselves kind of unravel, to be honest, I think our perception is a bunch of this was associated with the just absolute scarcity of PPE, right? We're not, what we're doing, what our researchers are doing, they're not directly exposed to patients with infectious disease. This is the same problem that would be if research labs were at a shopping mall, right? It's just straight hygiene procedures, cleaning, maintenance, and those kinds of things, and changing kind of how we do things. Shaking hands and those kinds of things all has to be sorted out. But I think the universities, I mean, they do this for a living. They've sorted out remote teaching. I'm sure we'll get the rest of this sorted out in due course.
Fantastic. And are you still getting a good number of inquiries about the technology coming in?
Yes. And in fact, the COVID stuff has stimulated conversations. There are a lot of people who kind of instantly saw the kind of synchronicity between what we can do and what has never been done. I think it is, we'll see how it all plays out. But yes, numerous people calling from several new corners of the world, kind of which could expand our footprint. Very exciting to see how that might all play out. But pursuing them all with our normal intensity.
Richard, thank you very much for taking the time to talk to us.